Good afternoon to everybody. Thank you for being with us for the MD Anderson Non-Coding RNA Center seminar. And first of all, I have to thanks to all of you who are here with us, because without attendees, all these webinars make no sense. And of course, I have to thanks to the people who organized all these things for all the RNA centers in the United States and Canada. And also thanks to the speakers. They are young and bright people who will tell us today about interesting findings on non-coding RNAs. We are here in MD Anderson Center of Non-Coding RNAs, who are moving forward from discoveries of different type of non-coding RNAs, basic science, description of these non-coding RNAs, identification of function, and very important, we move these discoveries toward the biomarkers and toward the clinical trials quite in a very quick way. Today there are two speakers, both are fellows of the Non-Coding RNA Center, Dr. Simone Anfossi and Dr. Christian Rodriguez Aguayo, and both of them will present you exactly what we told you. They will present all the panel of discovery till clinical trials. The center has a lot of collaborators. It's led by Dr. Anil Sudet by myself, with the co-lead of Gabriel Lopez Berenstein. And basically what is the main goal of the center, except promoting non-coding RNA, it's promoting young scientists and bright scientists in their career. You will see both of them are basically foreigners who come in the United States a long time ago. They have done PhDs, postdoctoral fellows, and now they are instructors and are basically fellows of the center. And because I don't want to steal the show, let's start with the first talk by Dr. Simone Anfossi. He basically will discuss about the fundamentals of interesting new non-coding RNAs. And in fact, he has a very fancy title, and looking forward also for a fancy talk. Simone, good luck.
Today my talk will focus on two aspects: the role of non-coding RNA inside the cell. Mainly we'll focus on Pyknons. They are a newly discovered, highly repeated sequence, and specifically we'll focus on Pyknon 19 that is located in a primate, a human-specific non-coding RNA identified in our lab. And also I will talk about the role of non-coding RNA outside the cell, specifically the role of microRNA included in extracellular vesicles secreted by tumor cell in the regulation of tumor-associated neuron. This was done in collaboration with Jeff Myers. As I said, most of the genome doesn't code for protein. It's a non-coding sequence, which is the function of the Pyknon. So the Pyknon name comes from the ancient Greek words. It means dense and frequent. And why this? Because these Pyknon sequences are non-random pattern of short repeated elements. They are consisting basically on 16 nucleotides that occur in the genome in the intronic and intergenic region at least 40 times, and at least 1 time in the exonic region. This Pyknon was discovered by collaborator Dr. Kelly. Dr. Gautsos in Jefferson University is a Greek guy, so this is why he gave this name, a Greek name, to the Pyknon. So he identified that there are more than 100,000 distinct Pyknon identified in human genome, in intergenic and intergenic regions. And more than 200,000 distinct Pyknon are present in the coding region and a non-coding region, 5' and 3'. They are present in more than 30,000 transcripts that derive from roughly 20,000 human genes. And as you can see in this cartoon, the Pyknon can be present in the exon, in the intron, in ongenic region, and specifically they are enriched in a 3' UTR sequence of the gene. And here you can see an example of a Pyknon present in the 3' UTR of the PIK4 gene that code for XIAP, inhibitor of apoptosis gene. And you can see here that there are more than 95 distinct Pyknons in this sequence, and 22 of them are also present in other UTR of 9 genes. And you can see in highlight in red, in yellow, and in green, our Pyknons that are present in this UTR sequence at least 2 times. You can see TCGA, this is in red, these 2 in yellow. There is these 2 sequence, and in green there are these 2 sequences. The other Pyknon in gray are present only once, but as we said as a definition, they are present also at least 40 times in the genome, so they are present in another aspect. And interesting that the space between the 2 consecutive Pyknons are roughly skewed to 22 nucleotides, so they are pretty close each other. So which is the characteristic of the Pyknon? 1/3 of the Pyknon actually can interact with the neighbor region, mainly 60, 80 nucleotides, and this interaction allows the RNA to form a secondary structure like hairpin-shaped structure. And everybody knows that secondary structure, hairpin, is important for the functional characteristic of the RNA. And Pyknons are highly enriched or depleted in the 5' UTR, coding region, and UTR of genes that regulate biological process. Here on the right, there is a short summary of some of the functions that are regulated by the gene with the Pyknon inside. And the complete list, you can find it in these 2 paper from Dr. Ribowitz's group. One important thing to stress is that Pyknon are genome specific, which means that mouse Pyknon and human Pyknon are different. There is no conservation. And even if they are enriched in introns that regulate the same process and molecular function, the Pyknon of human and mouse on that region are completely different. Another thing important to notice is that the Pyknon share some similarity with the microRNA. In particular, 40% of microRNA has similarity with this, almost 700 Pyknon. So based on this evidence, we hypothesize that Pyknon may have some gene expression or regulatory function. So we want to see if this function can be also present in the cancer, have some function in regulated cancer. So in our lab, we perform a gene expression profiling using a customized array that was developed in our lab. We found that several Pyknon are highly expressed in the cancer compared to the normal tissue. You can see here Pyknon 14, 40, 41, 44, and Pyknon 19. We focus mainly on Pyknon 19, as we found that highly expression of Pyknon 19 in colorectal cancer was associated with a poor prognosis. And we see this in one cohort of patients and in another cohort of patients. And particularly, we found that Pyknon 19 sequence is present inside the sequence of the newly discovered human-specific and primate-specific low non-coding array called N-BLR that was identified in our lab. And this you can see in red, the Pyknon 19 sequence. When we measure the expression of N-BLR by in situ hybridization performed by Dr. Zhang, a former member of RNA coding, now is in Indiana University, you can see that in normal tissues, N-BLR is a weakly expressed. You can see here also in higher magnification. Where in the tumor tissues, N-BLR is highly expressed, but you can see that is not expressed in the stroma, so it's specifically expressed in the tumor. And you can see also here at a higher magnification. And the distribution of N-BLR is both nuclear, you can see a dot, and also cytoplasmic. You can see also here in the stroma is not present. Also here we have a red blood cell that doesn't express present. So we want to measure and quantify the expression of N-BLR in the tissues. We perform this with the help of Dr. Burks, that is co-director of the Cell Core Imaging Facility at MD Anderson, and we found that adenocarcinoma and metastatic tumor express higher level of enabler compared to the normal tissues. And also importantly to note that also benign and colitis colon tissues has a comparable level of enabler compared to the normal tissues. So it means that enabler is specifically overexpressed in colon cancer. In the colon cancer, tissue. Importantly to know that Pyknons sequence partially overlap with the binding site of microRNA 200C. And also, Pyknon 90 is a 5' to the binding site of another microRNA belonging to the same family of 200 microRNA. That is near 141. So it means that enabler is able to interact with two microRNA and possibly regulating the expression level of these two microRNA. Indeed, when we overexpress enabler in RKO cell, we measure a decrease in the level of near-200C and near-141. It means that enabler can interact with two microRNA and possibly sequester a free copy or free molecule of this microRNA and inducing their degradation. Conversely, when we knock down enabler by siRNA, we have an increase of both two microRNA, near-141 and 200C. So we want to test if this inverse correlation is also present in vivo. So when we measure the expression of enabler, as we saw before, is weakly expressed in normal tissue, but it's highly expressed in adenocarcinoma. When we measure the 200C microRNA, we see the inverse correlation. So it's a high in normal and a lower expression in adenocarcinoma. And here we quantify by imaging analysis. Same thing with the near-141. When enabler is low, when near-141 is high. When enabler is high in adenocarcinoma, near-141 is low. And here you can see the quantification. So since near-200C and near-141 regulate EMT, we want to see and assess if enabler is able also to regulate EMT phenotype through the regulation of these two microRNA. And actually, we perform a macro array gene expression profiling in two clones that are lower expression of enabler due to siRNA. And you can see here two markers of EMT. E-catenin is a marker of epithelial phenotype. It is increased when enabler is knocked down. And bimentin is a marker of EMT, epithelial mesenchymal transition phenotype. You can see a decrease. So when enabler is knocking down, the two mirror level are increased, and we have a decrease of mesenchymal phenotype of the cell. The gene expression analysis was performed by Dr. Ivan in our center. And also, as you can see here by immunofluorescent analysis, when we knock down enabler, we have an increase of expression of E-catenin. So the cell acquire more epithelial phenotype. And we have a downregulation of two markers of EMT, bimentin and ZEB1. ZEB1 is an important transcription factor that regulates the acquisition of AEMT and mesenchymal phenotype of the cell. Also, you can see here at protein level, when we knock down enabler, we have a decrease of ZEB1 in these two different clones. And so, since EMT is associated with increased migratory and invasion ability of the tumor cell, when we knock down enabler, we measure a decrease accordingly, decrease of invasion and migration cell. So we want to see and assess which is the function of the Pyknon inside the enabler transcript. So we established two scrap transcript with the wild type Pyknon and one enabler transcript with the specific deletion of the only Pyknon sequence. So as we showed before, the overexpression of wild type enabler induced the downregulation of 200C, where the deletion of the binding site of 200C reverse the level of mid-200 similar to the empty vector. Accordingly, here when you overexpress enabler the wild type, if we have a decrease of 200C level, but the deletion of the enabler prevent the ability of enabler to induce a downregulation and decrease level of the mid-200C. I'm sorry, the enabler with the deletion of Pyknon. So accordingly, the deletion of the specific deletion of Pyknon in a wild-type enabler inhibit the ability of enabler to increase the migratory invasion property of the cell. And making immunofluorescence analysis with the help of Dr. Mani in a translational molecular pathology, we saw that overexpression of a wild-type enabler actually induces downregulation of E-cadherin, but that the specific deletion of Pyknon revert the phenotype to the epithelial phenotype. Also, you can see here the same as we saw before. The level of ZEB1 and Bymantin are increased by overexpression of wild-type enabler, but actually they are decreased when we remove and delete this Pyknon 90 sequence within the wild-type enabler. And also with the help of Dr. Rodriguez Aguayo, we want to test if the ability of Pyknon 90 to regulate the development of phenotype also is translated in ability to regulate the metastatic property of this. And actually, when we overexpress enabler, the wild type, we can see a massive metastatic lesion in the liver and also higher proliferation index. While when we overexpress the enabler with a deletion of the Pyknon 19, we have the similar result of the empty vector. So Pyknon 19 means that can regulate the ability of the tumor cell to metastasize to the liver. Since MIR200C also regulated the level of CIAP, that CIAP is an important anti-apoptotic protein, we want to see if the MIR200C also is responsible of a response to chemotherapy. Actually, as we saw before, overexpression of wild type enabler induces downregulation of 200C, an increase of his target accordingly. And to confirm that MIR200C is targeting CIAP, the overexpression of 200C reduce the messenger RNA of CIAP and also apoptotic. So overexpression of enabler induce downregulation or lower expression level inside the cell of MIR200C and increase of CIAP. CIAP was discovered to be associated with the resistance to 5-fluorouracil. So we want to test if the overexpression of enabler actually can regulate the response to 5-fluorouracil. And actually, even if you have a slight increase, we have statistical significant increase of resistance to the fluorouracil treatment when the cell overexpresses the wild-type enabler. You can see also into clone. Conversely, when we overexpress MIR200C by MIR200C mimic, and so induce a downregulation of CIAP, we have an increased sensitization of colon-20 colon cancer cell line to the treatment of 5-fluorouracil. So to summarize this first part of the talk, the overexpression on the lab of enabler, so according also the Pyknon 19 sequence within his sequence, we show that the MIR200C and luman 41 are sequestered by this non-coding RNA and induce the lower expression level within the cell of these two microRNA. And since these two microRNA is targeting ZEB1 increase, and ZEB1 is an inhibitor of E-cadherin expression, so the cell lose the epithelial phenotype and acquire more mesenchymal-like phenotype, which is associated with increased migration, invasion, and metastatic invasion. At the same time, the decreased level of MIR200C induces an increased level of CIAP inhibitor of apoptosis, what is associated with the decrease of apoptosis and response to 5-fluorouracil. And these two aspects were responsible of a poor prognosis. Furthermore, interesting to know that mid200C also is targeting PD-L1, that is important checkpoint inhibitor, and it is highly studied over the past years. And we didn't check this in our study, but it's a subject of further investigation. So increase the level of PD-L1 due to the increased level of mid200C is responsible of a checkpoint inhibitor and immunoescape by the tumor cell. And also this can be can play a role in determining the poor prognosis. As I showed before at the beginning of the presentation, Pyknon 19 is associated, high level of Pyknon 19 is associated with poor prognosis. And since one of the mechanisms of Pyknon 19 we highlighted here, but Pyknon 19 is expressed by definition more than 40 times, is present more than 40 times in the genome, it's possible that alternative or additional regulatory function can be played by Pyknon 19, but this subject is still under investigation. Also important to notice that Pyknon compared to the microRNA are much more abundant, like 64-fold higher. So it's possible that Pyknon may play a significant role in the regulation of a biological function of the cell. So let's shift the gear and let's talk about the role of extracellular vesicle microRNA in regulating the tumor-associated neuron phenotype. Over the past year, has been increasing interest and evidence that peripheral nerves can control the proliferation and progression of tumor cell, mainly by regulating immunocell and stromal cell, and also directly the neurotransmitter can work as a growth factor for the tumor. And also neurotransmitter can regulate neurogenesis, so this process make the tumor cell invade the peripheral and the neurovasculature, and so can drive the progression to metastasis. On the other side, tumors can secrete a neurotrophic factor that can induce the proliferation and a regulation of the function of the infiltrating neurons in the tumor. Actually, infiltration of neurons in the tumor tissues was found in several cancer types like prostate, gastric, pancreas, breast, ovary, and glioma. And the neurotransmitters secreted by the neurons can stimulate the tumor cell to proliferate and become more aggressive. The major role is played by adrenergic receptors. So norepinephrine or noradrenaline secreted by sympathetic fiber that are associated with the tumor can activate the adrenergic receptor signaling and activate a bunch of downstream signaling pathway that end up with the increased secretion of pro-inflammatory cytokine, androgenic factor, metalloproteins that help tissue invasion, and EMT regulator factor, and inhibit some immune molecules that regulate tumor immune response. Also, norepinephrine and epinephrine from the circulation can do activate the adrenergic receptor in the same way. And also tumor-associated immunocell can be regulated by noradrenaline, particularly like macrophages can secrete more pro-inflammatory cytokines, so they can drive the progression and the growth of the tumor. And particularly, it was shown that the use of beta blocker in a triple-negative breast cancer, patients that receive beta blocker, that is a drug used mainly specifically for heart disease, patients that receive beta blocker had better prognosis in terms of relapse-free survival and overall survival. So it means that beta adrenergic signaling can play a significant role in cancer progression. And so in our group, in Dr. Kalin's group, in collaboration with the group of Jeff Myers, is the chair of head and neck surgery, and the research was conducted by Dr. Amit and Dr. Takahashi, are the first author of the paper. we found that p53 status can regulate the differentiation and the phenotype of tumor-associated neuron, particularly the wild type p53. The tumor can secrete extracellular vesicles that are highly enriched in miR34A. And miR34A block the proliferation and reprogramming of the tumor, of the neurons. So the tumor-associated neuron maintain a sensory phenotype. On the other side, the mutant p53 tumor secrete extracellular vesicles. They are lacking or express very low level on miR34. So this induced the transdifferentiation of the tumor-associated neuron in a neuroadrenergic neuron. So they secrete higher level of noradrenaline. And we saw in the previous slide that noradrenaline can play a role in driving tumor progression. And actually, we found that in the p53 tumor deficient grow faster, but the treatment with the beta blocker actually slow down the tumor. So let's see in more detail what we found. So p53 null tumor cell in a xenograft model, we found that the neurofilaments are highly enriched in this xenograft model compared to tumor cell that has a p53 wild type. And also you can see here in immunohistochemistry analysis that tumor cell with p53 null has highly enriched of fiber and neuron filament compared to tumor cell that have a wild type p53. And actually tumor with the p53 mutated or null grow bigger and faster than a tumor with a p53 wild type. In another model, that the tumor grows spontaneously, specifically is a conditional knockout of p53 in an epithelial cell, and the mice were treated with a cancerogenic compound. After 28 weeks, we can start to measure a premalignant lesion, and after 38 weeks, a completely differentiated tumor. And you can see that tumor with p53 mutated is associated with an increased number of the neuron filament in tumor tissues compared to the tumor as a wild type. You can see at early stage a preliminary lesion and also at later stage. And so in patient, what happened that patient that has high infiltration of the neuron in the tumor, so high neural density, has a poor prognosis. So this is important that confirm the role of neural infiltration in the tumor. And also patient that has a p53 mutated tumor has a higher neural filament enrichment and density in the tumor. So to find what is the driver and responsible of this phenomenon, we make a co-culture of cancer cell with the neurons. And as you can see that cancer cell with a p53 mutation has a higher number of neural filament compared to keratinocyte and p53 wild type. And you can see in this picture on the co-culture in vitro that p53 mutated tumor cell induce a highly and massive branches and enrichment of neurofilament. And also we demonstrate that extracellular vesicles from a tumor cell are responsible for this phenotype. And you can see that extracellular vesicles from a p53 mutated cell induce a massive branches and packing of the neuron filament in the co-culture in vitro compared to the extracellular vesicle from a p53 wild type tumor. And also you can see here the quantification. And here you can see that extracellular vesicles that are labeled with lipophilic type can be taken up by neurons, both EV from p53 wild type cell and p53 node. And importantly, that the content, the cargo of EV, specifically here we label in green fluorescent that stain the RNA. You can see the green dot inside the cell. So the neuron. So it means that the RNA cargo within the EV can be delivered inside the neuron. So when we analyze the microRNA expression profile inside the EV from p53 wild type to p53 mutated, we see that MIR34A was one of the highest differentially expressed, and you can see that here in p53 wild type and p53 mutated. And actually, MIR34A was the main responsible of regulating the phenotype changes of the neuron. So when we deliver EV from p53 mutated, you can see compared to the control, we see an increase of branches and changes on the phenotype. But this was prevented by transfection with 34. So it means that MIR34A play the major role in regulating the changing of the phenotype of the neuron. And also, as you can see here, HN30R tumor cell with the p53 wild type, when we knock down MIR34A by shRNA and we deliver this exosome to the mice neuron in a co-culture experiment, you can see that an increase of the neurofilament by EV with a lower level of MIR34. And also in vivo, in a xenograft model, the knockdown or 34A in HN30 tumor cell is associated with an increased level of neurons infiltration in the tumor. So when we analyze the effect of the EV on the expression profile of the dorsal root ganglion, we see striking differences in the expression profile, two different classes. And when we did the gene pathway analysis, we saw the adrenergic signaling and the gene that regulate tumor growth, morphogenesis, and synaptogenesis signaling pathway and neurodevelopment were differentially expressed. So we hypothesized that the delivery of EV from p53 now can regulate the phenotype of the neuron. And actually, this is what happened. So in the previous model we showed before of p53 conditionally knocked down in the tumor, the absence of p53 was able to induce increase infiltration of neurons with adrenergic phenotype. Tyrosine hydroxylase as a marker of adrenergic neurons because it's an enzyme that is involved in noradrenaline biosynthesis. So high level of TH enzyme is a phenotype of adrenergic. And you can see also in terms of number and in terms of density, the p53 mutated phenotype of tumor cell was able to induce a highly enrichment of a neuroadrenergic phenotype. And importantly, in a p53 null xenograft, the delivery, orthotopically delivery of EV from wild type cell were able to reduce the presence of neurons with the adrenergic phenotype. And this phenotype was reverted by the administration of EV from p53 node. And also we saw this also in human neurons, and we saw that the delivery of EV from p53 node, so it means to have a low level of MIR34A, were associated with increased number of neurofilament and increased secretion and production of a noradrenaline. So it means that the neuron acquire a noradrenergic phenotype. And to confirm the role of MIR34A, we use EV derived from tumor cell where MIR34A were knocked down, and we co-culture with mouse neuron. And you can see here, similar to the previous experiment, the presence of neurons with adrenergic phenotype is significantly increased. And also the secretion of noradrenaline, production of noradrenaline is highly expressed. To demonstrate the innervation of the tumor actually play a role, sensory neuron was surgically ablated, and then the mice were injected, inoculated the tumor cell, and then the tumor were evaluated at 26 days. And you can see here that lingual denervation, so the removal of sensory fiber, reduce the ability of the tumor to grow. And importantly, the lingual denervation prevent the differentiation of the tumor-associated neurons to differentiate to adrenergic neurons. So in order to demonstrate that this is not due to the previous presence of adrenergic neuron, we pre-treated the mice in order to chemically ablate the adrenergic fiber. You can see here that the treatment reduced almost to zero the presence of adrenergic neurons, but the presence of the tumor actually induced a significant increase of infiltrating neurons with the adrenergic phenotype, and also accordingly the level of noradrenaline is highly expressed. This means that the presence of the sensory neuron in the tumor side are induced to differentiate to adrenergic by the presence of tumor with p53 mutation. And also here, to demonstrate the role of miR34A, when we knock down miR34A in the p53 wild type tumor cell, and we did xenograft injection, in absence of denervation, we have an increase of neurofilament associated to the tumor, an increase of neurons with adrenergic phenotype, an increase of noradrenaline secretion. Whereas the denervation, surgical denervation, prevent the development of this phenotype and the production of noradrenaline. And most importantly, in a two different xenograft model with p53 mutated or p53 knockdown, the treatment with beta blocker, carvedilol, significantly decreased in both two models the tumor's growth. This means that blocking the beta adrenergic stimulation by noradrenaline have an effect on tumor growth. And this can be important for patients that has highly infiltration of neuron with adrenergic and phenotype. So that you can see here, they have a worse prognosis to the patient that has lower infiltration. So the treatment of this patient with a beta blocker can be potentially highly beneficial. So to summarize this work, we show that a p53 status can induce the downregulation or lower expression level of miR34A in the cell, and accordingly lower level of miR34A loaded inside the extracellular vesicle. And these vesicles, when they are delivered to the neurons, are able to induce a transdifferentiation from sensory neuron to adrenergic neuron. So these neurons secrete higher level of noradrenaline that work as a growth factor for the tumor that grow faster and is responsible of poor prognosis. And so this is what we saw, that proliferation, progression, and prognosis are positively regulated by beta adrenergic signaling, while blocking the beta adrenergic signaling can be beneficial. In terms of therapeutic approach, beside the blocking the beta adrenergic receptor, can be a future direction of this project, try to inhibit or block the secretion of extracellular vesicle from a tumor cell, or try to block the secretion of noradrenaline or other growth factors secreted by a differentiated neuron to prevent the stimulation of a tumor cell by noradrenaline. And also, importantly, noradrenaline is can downregulate the effector function of immune cells, so can create an immunosuppressive tumor microenvironment, so it prevents the tumor immunity. And also, potentially, tumor cell, the expression of beta adrenergic receptor can be also targeted directly on the tumor cell side. So this is the end of the presentation, and as you can see, our lab is pretty big, and these are all the collaborations. So it means that these two work require the collaboration with a lot of people, especially thanks to Dr. Maya's group, Dr. Amit, and Dr. Takaichi, that performed the study of the neurobiology, and Dr. Rivus for the Pyknon, Dr. Coppes for the providing a patient sample for the Pyknon story, and a bunch of collaborators inside MD Anderson, biostatistician, and also collaboration with other institutions for the MIR34A and neuron story. And of course, thanks to Dr. Mani and Dr. Lopez, Dr. Kelly, and Dr. Sud that are co-director of our non-coding sector.
Thank you, Simone. Thank you very much.